Today, we wanted to thank all of our attendees who have decided to click through and watch this very exciting press conference unfold today. I'm Rebecca Vogt from Roswell Park Comprehensive Cancer Center. Now today we are very pleased to be announcing a new collaborative research initiative. Today you'll be hearing from the people who will be leading this important new project, and that includes Dr. Kunle Adunzi, the Deputy Director, the Robert Ann and Lou Wallace Endowed Chair in Cancer Immunotherapy, Executive Director of the Center for Immunotherapy, and the Chair of Gynecologic Oncology at Roswell Park. We also have Dr. Carl Morrison, Senior Vice President of Scientific Development and Integrative Medicine at Roswell Park, Dr. Hans Cassignol, Executive Vice President and Chief Clinical Officer, Physician Executive at Catholic Health, Dr. Jean Morse, SUNY Distinguished Professor, School of Pharmacy and Pharmaceutical Sciences, and Director of the Global Virus Network Center of Excellence at the University at Buffalo, and Andy Felton, Vice President of Product Development, Clinical Next Generation Sequencing, and Oncology at Thermo Fisher Scientific. Now, each of these individuals will have just a few minutes to discuss their role in this initiative and what they will accomplish together. To note for members of the media, if you have any questions during the presentations, absolutely feel free to use that chat button down below on the screen to ask them. We will get to them during a question and answer session after our speakers, which we are hoping goes between 11:20 and 11:30. That all being said, I think we can get started. So I will pass the mic over to Dr. Adunzi. So Dr. Adunzi, can you please tell us a little bit more about the initiative that we are launching today?
Well, thank you. First, good morning, everyone, and thank you for joining us to announce what is truly a remarkable and an unprecedented collaboration. Today, we announced the Western New York Immunogenomic COVID-19 study, which is a really new initiative that unites 3 leading regional healthcare organizations in Western New York, as well as an industry partner, so that we can better care for patients with COVID-19 based on a better understanding of the disease, because COVID-19 is very new. There's very little known about it. So when COVID-19 first became so concerning, Dr. Carl Morrison and I got together. We were just exchanging ideas for what can, asking the question, what can we do to contribute to the world's understanding of COVID-19? In fact, maybe something that could impact and maybe even save lives. So we quickly realized that there was something we can actually do. And that not only is there something we can do, we actually have a moral responsibility to apply what we've been doing, what we've learned in cancer research at Roswell Park over the last 5 to 7 years. How can we apply this to COVID-19? As we all know, no two people are alike, and this is clearly being seen when it comes to COVID-19. Some individuals don't get sick at all. They test positive, but they don't get sick. Some get the usual flu-like symptoms. They have mild disease for a few days, and some become very sick and may even develop life-threatening symptoms. So our thought process is this: could we predict what group people will fall into? Which of these categories are people going to fall into if they come in contact with the virus? This is a huge undertaking, a huge question that is going to be instructive for the entire world. And we could not do this alone. So I am pleased to have several of our key collaborators with us for this announcement today. We are very grateful to our partners at the Catholic Health System, and they have been taking on a very important role in our community, treating so many local residents who are sickened by COVID-19. Dr. Cassignol will be leading this effort within the Catholic Health System. Dr. Jean Morse from the University at Buffalo, when we called him to discuss this idea, he also enthusiastically, without any hesitation, said, yes, just let me know how I can help. So thank you again to Dr. Morse, University at Buffalo, and to Thermo Fisher Scientific. They provided technology and the materials that we need to be able to do this work. Not only have they provided some of the cutting-edge precision medicine technology that they have also helped to defray some of the costs of this research. And so as we announced the Western New York Immunogenomic COVID-19 study, we believe that this is one of the first precision medicine initiatives focused on this deadly disease. And we also believe that we can limit its deadly impact by marrying thoughtful strategy to next generation sequencing technology. And this is an opportunity that we never had before with any previous pandemic. And using this technology in a few short years could really change how we detect and diagnose COVID-19. It's helping us in cancer. We believe we can deploy it also in COVID-19.
Fantastic. Thank you so much, Dr. Adensi. Now we're going to switch over and pass the mic once more over to Dr. Morrison. Dr. Morrison?
Thank you all for calling in this morning. What I'd really like to do is, without being too technical, try to explain to you from a clinical perspective, what is it we're trying to do that's different from what everyone else is doing, right? And I think that if you stop to look at what primarily is the testing that's going on today in COVID-19, you see that there's two approaches, right? One, which is the most common approach, is to look for detection of active infection in an individual by either testing their nasal secretions or the blood, right? And even if all these tests were perfect, which we know by now that they're not, the problem with this approach is that test is only valid for the day the sample was taken, right? A person could have a negative test for infection one day and a week later be exposed and become an active spreader of the disease, and all the while think he or she is not infected. And it's totally impractical to think that we're going to test the entire population on a weekly or a bi-weekly basis for infection, right? So others, and the second approach that you see for COVID-19 out in the public, right, is to look for evidence in a person's blood that they've previously been exposed to COVID-19. And primarily what they're looking for are these proteins in the blood called antibodies. And these antibodies, which can be very specific to COVID-19, if present, would indicate that a person has been previously exposed to COVID-19, and they're likely not to get sick upon re-exposure. And while this is a very important approach, right, one of the major problems is today the vast majority of people have not been exposed to COVID-19, and this approach, like the first approach, is really only valid at the time the testing is performed for patients that don't have antibodies in their blood. So we took a little bit different approach to this and said, well, okay, well, if antibodies in the blood do have value, right, and we know there's some problem there, why don't we look at the cells, what they call immune cells, that are responsible for making these antibodies? And these cells, called T and B cells, are in your blood. They are your defense to all pathogens, virus, bacteria, and to cancer. And that's why our expertise in cancer has allowed us to apply some of this technology to COVID-19, right? And there's millions of these cells in your blood. And each one is unique, right? And a subset of them, if present, have the capability to respond to COVID-19 and fight off infection, right? And this goes back to what Dr. Adensi was saying, right? That we know that some people don't get sick at all. Some get flu-like symptoms and are over in a few days. Some become very sick and even die, right? And so our study is focused on the fact that if we could look at these B and T cells using some advanced technology, right, that we could predict if you become infected or get exposed to COVID-19, who's not going to get sick, who's going to get usual flu-like symptoms, and then who is potentially is going to become very ill and possibly even die, right? And this is very much, right, what we do in cancer every day, looking for these specific mutations in genes we call finding the needle in the haystack, right? And we do that through the technology called next generation sequencing. And thanks to groups like Thermo Fisher Scientific, right, we're able to take the same technology with new reagents that they have produced specific to these B and T cells and take the same approach and apply it to COVID-19. So in the end, our goal is to develop a blood test that a patient would have one time, or at least not more than once every 6 to 12 months, and would say, if you were exposed to COVID-19, you may not get sick at all, or you may get the usual flu-like symptoms, or you could die, possibly even die from this disease. So if we know how to do that, it could have benefits not only from a health standpoint, but also from an economic standpoint. It could be the key to trying to say, how should we reopen the economy, right? For those people that are not going to get sick when exposed. So, I'd like to say that we're very excited to get this started work and improve the lives of people with exposure to COVID-19. And we thank you very much for your time today.
Fantastic. Thank you so much, Dr. Morrison. We are now going to hear from Dr. Cassignol from Catholic Health. Dr. Cassignol.
Thank you again all for joining us today. I thank Catholic Health to say that we're excited to partner with Roswell Park, University of Buffalo, and Thermo Fisher would be a great understatement, and specifically because of their research background and the quality that they bring into the field. We pride ourselves equally in terms of the quality of care that we deliver to our patient population in Western New York. We believe we actually are some of the leaders, if not the leader in the area. That is really has been exemplified more so by the way we really re-engineer ourselves over the past 7 to 10 weeks or so, creating a treatment center specifically dedicated toward COVID-19 at our St. Joseph campus. Catholic Health as a whole have really taken a significant amount of effort and energy with the capacity at which we focus the way we've treated our patient in terms of the COVID-19 population as a whole. Having partners that will help us increase our capacity to predict who's actually is going to suffer the brunt of this disease will be truly monumental in the task that we have ahead for us, not only as a community, but also as a world as a whole. One of the biggest thing that we've actually noted as a healthcare system, the disease does not spare anyone, and it does spare just about everyone at the same time. There's not really a very big predictor from our own experience as we've seen for the people we've treated in our system since this started. We've seen if loved ones, family members of our community entered our institution with a way that seems pretty much consistent with a flu-like illness. But yet, at the same time, there's no way to predict whether those loved ones, family members, community members will actually be walking out within 2 or 3 days or actually end up in the ICU setting. And really what we need something that's going to tell us who of those community members, family members, loved ones that actually would be walking out in a couple of days and who's going to be literally needing intensive level of care within the ICU setting. Because our own experience, just like the world experience, getting into the ICU is the one thing that we don't want for any of our family members or community members within the disease process, because that's when really the brunt of this disease really take a toll in our community. I can't really repeat anything more than what Dr. Odansi and Dr. Morrison said in terms of both the human and the economic impact this has done. We're really looking at 3 things specifically. We need to do better for our community in terms of prediction-wise. We need to do better for our caregivers in terms of giving them more tools that they have right now. And this is just an added tool that we could give them in terms of giving more capacity to test, give them more capacity to protect themselves as a whole. We think we're the ideal partner because what we've done in our community, we've treated close to half, if not more than half of our COVID-19 population in Western New York. And we're really, really excited, to say the least, to actually take partnership of this. And last but not least, our caregivers, when they actually heard within the institution that we'll be taking our partnership in such a research trial with names such as Roswell Park, University of Buffalo, Thermo Fisher, to say they were excited would be a gross understatement. Thank you again for joining us.
All right, thank you so much, Dr. Cassignol. Now we'll shift on over to UB and Dr. Jean Morse.
Am I unmuted? I am. Okay, thanks everyone. Great to be with you all today. I think many of you may know me since I've been here since the early 1980s when AIDS was first introduced to Western New York. And over the last 5 years or so, I've been working very closely with Roswell Park to build translational research and drug development collaborations. The other thing we've done over the last 5 years is become a center of excellence for the Global Virus Network. This is a network of virology research centers around the globe. I think there's about 40 centers now. And when we became a member, in 2017. Our application was co-directed by Dr. Erwin Gelman from Roswell Park. That network, the Global Virus Network, is led by Dr. Robert Gallo, who's the scientific director. Some of you may remember he was here in Buffalo a couple of years ago when we had our formal launch of the center. That group gives me the opportunity to have weekly conference calls now during the COVID pandemic and really have a finger on the pulse of what everyone around the world is trying to do, those in countries that are facing the outbreak now, and those in developing countries that are waiting for the epidemic to come. In February, the GVN formed a COVID-19 scientific committee that I was a member of. Dr. Igor Kuzanov from Roswell Park also was on that committee. And as a result of our activities there, a number of different trials have come to Buffalo that we've been introducing over the last month. I also wear a hat with the SUNY Global Health Institute, and that is a network of the academic health centers within SUNY, forming partnerships with global partners. And in that effort, I also work with Dr. James Moeller, who's the Chief of Interinstitutional Academics at Roswell Park. So there have been a number of collaborations, a number of projects that we've been working on both in the US as well as with global partners. A very interesting aspect of this project is the effort that has been going on to build academic business partnerships. So the opportunity to bring together clinical sites where patients are being cared for, leading research programs, and also leaders in our community in scientific business development like Thermo Fisher is also a very exciting part of this project. So I think what's very exciting about the project is that we have an opportunity to do something very unique here. We're also connected throughout the state with the other SUNY health centers, and we have collaborations with our affiliate centers. The GVN Affiliate Center for UB have centers in Kingston, Jamaica, as well as Harare, Zimbabwe. So many of the things that we'll be doing, we'll be able to introduce there as well.
Excellent. Fantastic. Thank you, Dr. Morse. And then we are going to shift over to Thermo Fisher Scientific and Dr. Andy Felton.
Here and meet you all. Thermo Fisher team is very excited to partner with the Western New York Immunogenomic Consortium and University of Buffalo and Catholic Health as the provider of the underlying technology that the team is going to do the research on. So I'm going to give you a little bit of a view of what we're going to provide to the consortium in terms of the technologies and how they work in a very brief and hopefully very concise manner. So Thermo Fisher Scientific is one of the largest providers of scientific and technical in the world. We're one of the major manufacturers of the current COVID-19 screening test. But in this case, what we're providing to the team is a set of technologies for next generation sequencing. That's sequencing of the individual base components, in this case, of the immune system itself. So we're utilizing a technology called the Genexus next generation sequencer. And the major advantage of that, it was just launched at the end of 2019, is its incredibly rapid turnaround time. We can do the whole process in as little as 14 to about 24 hours with very minimal hands-on time for the technicians who are going to go do the work. So about less than 10 minutes of hands-on time in total to set up the instrument and then wait for the results at the end. System also provides integrated data analysis to make the interpretation of the data at the end of the process very simple and straightforward. In terms of the assay components we're providing to the team here, there are two elements of that, the Oncomine T and B cell receptor sequencing assays. So those are assays that provide sequencing of the immune repertoire, and the immune system, as I'm sure many of you will realize, is incredibly complex, very diverse, and requires a very systematic and technical approach to sequencing it, but we've done that in a very easy fashion. So the whole process can be completed, as I said, in about 10 minutes. the immune system has really been a dark corner of the genome, if you will. In the original genome sequencing efforts, it was one of the parts that was really left out of the process. And these two brand new assays really help us understand the general complexity and how the immune system is changing in response to this really challenging disease. So we're very excited to partner with the team here, and we look forward to generating some really fantastic data and hoping to really change the paradigm in healthcare and improve the human condition. Thank you.
Wonderful. Thank you so much, Dr. Felton. The explanation, super helpful. Now I'm just going to switch back over to Dr. Adunzi. He's going to say a few words just to sort of wrap it up before we get to question and answer session. Dr. Adunzi?
Once again, I am just so grateful about this, that we have this incredible team, the Catholic Health System, Dr. Cassino, and Dr. Jean Moss from UB, and the Felton from Thermo Fisher. This is a tremendous team, and everyone brings tremendous asset to the table and to our community. I must also thank another very generous, important partner, the 11-Day Power Play. This is a nonprofit that gets our whole community united around hockey every summer to raise funds for Roswell Park and other organizations that help cancer patients. We're very proud and grateful to announce that our Western New York immunogenomic COVID-19 study has been launched with a $150,000 gift leadership donation from the 11-Day Power Play as part of the Roswell Park's COVID-19 Response Fund. We're very grateful. This is a needed vital support, and because we expect the cost of this study will be up to about $1 million. And the pace and scope of our efforts will be dependent on community support of this COVID-19 Response Fund. So the question is, why is this work so vital? I think everyone has spoken to this, but I'm going to make the point once again. What might we accomplish from this work? We have the unique opportunity to not only improve the outcome of patients who will get the most serious cases of this disease by directing us to intervene earlier with more careful monitoring and intervention, but also to enable the most effective, efficient use of PPEs, hospital staff, other essential resources that are so dangerously stretched with this global pandemic. And we hope that, like us, our community members will want to not sit on the sidelines, but to support an effort led by some of the premier experts in this emerging field of immunogenomics. So I want to thank you all once again for sharing the vision for the Western New York immunogenomic COVID-19 study. As so many of our colleagues have discussed here today, this is science and work that cannot wait. The findings that come from this study could save many, many lives locally and globally. And we will now move on to our question and answer session. Thank you.
Fantastic. Thank you so much again, Dr. Adunzi, and thank you to everyone, all of our panelists here today. So we are seeing a lot of questions pop up in our chat, and we thank you guys all so much for submitting them. Two things I want to announce before we get to it. We do have our press release about this announcement posted on our newsroom, that is at www.roswellpark.org/newsroom. And then we also wanted to share the link for those interested in possibly supporting this effort with a donation, and that is give.roswellpark.org/COVID-19. Again, give.roswellpark.org/COVID-19. I will start with one of the first questions that came through from Tracy Drury at Buffalo Business First. And I think this might be a good one for Dr. Casagnol to address. But again, will you be testing positive COVID-19 patients to help develop the test?
The answer is yes. The protocol have actually been really developed by Dr. Adunzi, so I can't take any credit for this. So I'm aware of the process, but don't give me any credit for the process. The process would be, as patients would come in our facilities, we'll go to the process of actually testing them to see whether they're a harbor of the virus or not. If they are a harbor of the virus, then at that time, we actually will be specifically draws which Dr. Adansi and the rest of the team that involved in this research will be doing the sequencing on. And again, repeated at another time interval.
Okay. A lot of similar questions sort of popping in. How does this study really get off the ground? I mean, do we need volunteers? What do we need? And I'll address that to anyone who would like to take that on.
Well, the recruitment site, which will be the St. Joseph campus, where we have the treatment center, will be the recruitment site. And again, technically, our EDs also will be part of that process in terms of depending where we want to initiate the blood draws in terms of what part of the disease process as a whole. One could not actually call and just be part of the study unless Dr. Adansi changed something drastically on the protocol. But one can call and actually donate if they want to support the study, though.
Dr. Dunsi,
Yeah, let me just quickly add a few comments. First of all, the protocol actually has been approved by the Institutional Review Board, both at Roswell Park and at the Catholic Health System. So the protocol is ready to go. We think we might be able to launch as early as quickly as next week, where we actually begin to collect these blood specimens from patients. Patients will be enrolled from Western New York region, the Catholic system, Roswell Park, partnership with the University at Buffalo. And the point is these specimens will all come to Roswell Park for all of these analysis that we've talked about. So this will really, because we have the Catholic system involved, remember the Catholics have a dedicated hospital for COVID-19. That really will help this project. Moreover, we have the opportunity of enrolling cancer patients from Roswell Park. So how does COVID-19 interact with cancer? Because both cancer and COVID-19 require the immune system to fight them off. What is the nature of this interaction? So there are additional lessons that we're going to be learning from this study.
Okay. You talked about, again, something as early as next week opening up so that people could start taking part. I mean, talking about timeline, we have a question coming in. When do you expect to complete the study?
I'll take that question again. This is a very rapid story because the COVID-19 story is that when you get sick, it's very quick. So this is not a study that's going to take years and years. Our goal is to potentially complete the study within a few months. We're looking at probably within a matter of about 6 months.
Okay. All right. One question that we are seeing pop through, is it possible to study the cells of someone who has passed away? Is that something that would be looked at?
You want me to take that one?
Yes.
Yeah.
It's very possible, right? And, you know, Andy Felton at Thermo could go into that further detail if we want, but yes, we can if the DNA is available, or the RNA, both.
Yeah.
Dr. Feldman, do you have anything to add to that?
Yeah, sure. What's required for the assay and the test to actually run is simply blood from which we extract the two nucleic acids that Carl just mentioned, the DNA and the RNA. And you know, if unfortunately the patient is deceased, as long as their blood sample is still available from them that were drawn before they died, we would still be able to do the test itself.
Okay. Now, the goal is to create a one-time blood test. I mean, what will these tests show and how will it be used? And I open that up to anyone who would like to take that on.
Well, you know, it's a bit early to say until we see the results how the test would be presented in the future. But you know, you would like to think that this is really what they call an algorithmic analysis, so that you have a risk score for people, and you would say that, you know, if you get exposed to this virus, your risk of having serious disease is X, versus someone else whose risk score of having a severe outcome is Y, and so forth.
All right. One more, I think, before we can wrap up, and that is this a study that is kind of, I mean, we always look to the future. That is what research is really all about. I mean, is this study going to be geared to dealing with more also like future pandemics rather than sort of the immediate understanding?
Try that one. I think certainly the whole thinking behind developing the Global Virus Network is that there will be more pandemics.
Yes.
And the nature of the acute outbreaks is usually that they kind of peak and go away. But even as we're discussing now, there may be additional coronavirus infections as part of this pandemic. So I think it's both. I think it's for now and the future.
Fantastic. We have hit the 11:30 mark. Time certainly does fly. We want to thank again all of our panelists for taking part, all of our attendees and members of the media who have decided to take time out of their day to watch our press conference. Again, the press release, we should be distributing it very shortly via email. It's also posted on our newsroom, roswellpark.org/newsroom. And again, there is also the link if anyone is who would like to possibly support this effort with a donation. That is give.roswellpark.org/COVID-19, which will also be in the press release as well. Thank you everyone for joining us today, and have a good rest of your Thursday and then afternoon. Bye everyone.
Thank you.
Thank you.